Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is obviously related to the DNA repair system, and this topic is the recombinational repair. Okay, before going to this topic, I want to say to the newcomers that please you subscribe my channel and share. If you seeing continuously my channel, so please you subscribe and share my channel for you can say the motivation of me, and obviously for getting the notification. If you don't subscribe this channel, if you don't share this channel, so the growth of this channel will be stopped, and for getting the notifications, obviously you after subscription you just hit the bell icon, and we all know that in case of subscription there is no needed of money, so please subscribe for getting the motivation to me. Okay, so this is the topic, the recombinational repair. So recombinational repair is what? That is, it's a process of filling a gap in one strand of the double-stranded DNA by retrieving a homologous single strand from another double-stranded DNA. That means here it is saying that when the parental DNA, that replication fork is formed like that. Replication fork is formed. So from this replication fork, from this strand one daughter strand form, and from this strand one daughter strand form. So what is saying that the process of filling a gap in one strand of the double-stranded DNA by retrieving a homologous single strand from the another double-stranded DNA. Suppose if there is any damages is occurred, so in that situation it is recovered by this strand. Okay, this is a simple word, and it is a obviously post-replication repair method. That means after completion of the replication, this repairing method is done. When DNA is replicated, the dimer that prevents the damaged site from acting as a template. We all know without a template, new DNA cannot be formed. So if any damage is seen in the, suppose this is the parental strand, and if any damage is seen like this side, so new strand that is formed without completion of leaving a gap like that. Okay, suppose again I repeat this line, if any damage is seen like that, the strand of the parental DNA, so the replication after replication the strand is formed leaving a gap in the damaged site. So this portion is repaired by this recombinational repairing process. So gap in newly formed DNA, whereas the other parental strand forms the normal. So there is a problem created, but this is the normal. So in that situation, the normal strand, double-stranded DNA formed here, and the gap opposite the damaged site is filled by taking the homologous single strand of the DNA from the normal duplex. So what is saying that if there is a gap formed, this gap is filled by taking the part of this newly formed DNA, that means other, that is normal duplex. So from this part of the newly formed DNA, the gap is filled, and following this single strand exchange, the recipient, and here this one called the donor and this one called be the recipient, and the recipient duplex has the parental damaged strand facing a normal strand. So this is the recipient one. So here I already explained that the parental strand is damaged, but the newly formed strand or the daughter strand that be not damaged, that is normal. So here the only main problem, the gap is remaining here. Okay, and the donor duplex has the normal parental strand facing a gap that can be filled by repair synthesis. Okay, after giving some some part of the newly formed DNA to this for the filling of the gap of this newly formed DNA. This part again repaired by the repairing system. Okay. So this is the this is the graph diagram. You can see. So here this is the double helix structure and replication is occurring. So in the first case you can see this is the damaging part. This portion is the damaged part, and that's why a gap is already formed here. But this portion is the normal because here the parental strand is normal. So how this gap is filled? From the normal copy. From the normal copy means daughter copy, okay, not the parental copy. From the daughter copy, the portion is cutted and just the portion is cut down and it is attached with the gap. It is attached with the gap, okay. This portion is cut down from here and attached with this portion to fill the gap. Clear? This is the first one. This is the second one. This is the first one, this is the second one. Okay, I think you can understand now properly. Okay, so if the damaged site cannot be cannot be bypassed, then synthesis of the daughter strand will terminate. So why this gap is remain here? If damaged site is there, so if the damaged site is not be bypassed by the newly formed daughter strand, then what will happen to the daughter strand? That it will be terminated. Okay, it will be terminated. In such cases, the replication fork stalls and reverses a short distance so that a duplex formed between the daughter strands, and the incomplete strand is then extended by the DNA polymerase using the undamaged daughter strand. So what is happening here? That the replication fork reverses the short distance, just reverses the short distance, and in that way duplex formed between the daughter strands and the incomplete strand is then extended by the DNA polymerase. So after this situation, the strand is extended by the DNA polymerase using the undamaged daughter strand. So damaged daughter strand is not applicable, not needed for the replication. Here the undamaged daughter strand is very important because here this undamaged portion, from this undamaged portion, the one strand part is cut and attached with the damaged parental strands, complementary strand, and form the perfect strand. So now what is this situation of what is happening in here? Now after fulfilling this gap or after completion of this strand, the gap is now try to fulfilling. Okay, the gap is now try to fulfilling, and the replication fork moves forward again by a process equivalent to branch migration step of the homologous recombination. As a result, damaged site is bypassed and replication continues. So, as the damaged site, that is totally okay, one second. Okay, so one thing is that this part, after replication, this part is totally cut off from this newly formed DNA strand for attaching this site. That's why here the replication reverses again. Here the replication reverses again to complete this gap. Okay, and there is no problem created. Only here the important thing is that the damaged site must be rejected, or the damaged site must be totally amputated. Okay. So this is the total recombinational repair system. And here important thing is that another important thing I have to explain in this repair system, that is, if a break in the phosphodiester backbone of the DNA, this is not repaired before replication fork passes. Okay, but this thing is another thing where the damaged base is already there in the parental portion. But this second part, this is the another thing. Here the break in the phosphodiester backbone, and this is not repaired before the replication fork passes. And the replication process leads to double strand break in one of the daughter and double helix. And the replication fork is lost. And this is called the replication fork collapse. So what is saying that? Again I repeat this line. If the break in a phosphodiester backbone of the DNA is not repaired, it is not repaired, by any cases it is not repaired before replication fork passes. So the replication process leads to double strand break. Okay, if it is not occurring, then the double strand break in one of the daughter double helix, and the replication fork is totally lost if the repairing is not completed. So in that situation, the replication fork becomes collapse. Okay, replication fork becomes collapse, and obviously in that situation the repairing system, this is the recombinational repair system, is utilized for the completion of the strand. But how this is possible, I will describe in the second part. Okay, so here when the replication fork try to be collapsed, in that situation the break can be repaired by the form of the homologous recombination. Here a single 3' stranded overhang is created, and reg A, it is, you can say, one kind of factor that mediates the invasion of 3' overhang into the donor homologous duplex, and donor homologous duplex that try to donate this 3' overhang to the recipient one. And the donor DNA duplex now acts as a template for the DNA synthesis, and that's why Holiday junction is formed. So cleavage of the resulting Holiday junction that be restored, and the replication fork that not to be collapsed. Okay, or you can say that replication continues. So this is the rough diagram where you can see that replication is occurring in the first stage, but there is a nick is created. Replication fork encounters a nick. In the second stage, what is seen now here, the strand invasion. That means one strand is cut off from the donor and inserted in the recipient one. You can see that. In the third stage, the branch migration cleavage of the Holiday junction. So here the branch migration, there is the Holiday junction is created, but in this stage the Holiday junction is totally broken down and branch migration is occurring. And you can see the last step here, the replication fork now restart. Clear? So this is the process of the recombinational repair, and I will try to explain another repair system, that is the SOS response and the repair of the double strand DNA break. So you will continue with my channel, and I will try to explain another topic related to the DNA repair in my next video. So thank you.